We're here to talk about the initiation of NCI-MATCH, which is approved, as I said. It is, as of today, launched. It's unprecedented in its scope. And it uses, most importantly, an integrated and validated diagnostic to define patient care and define our understanding of how we're going to move forward. The last thing I'm going to say before I introduce the people who really you should listen to is to say a few words about the going beyond this trial and how this trial fits into the President's Precision Medicine Initiative. We hope to, if we are fortunate enough to receive the resources that have been requested from Congress, to be able to leverage this particular trial as well as our other precision medicine trials and to develop further match-like studies in which we can go deeper into particular diseases based on the early signals we get from this investigation. We think that doing that will help change the entire paradigm for cancer clinical trials and will also allow us to collect through the biopsies that are a part of this program the kinds of materials that will fundamentally allow us to use clinically annotated information from those biopsies to get a much better understanding of how we go forward to understand both tumor heterogeneity and resistance to novel therapies. So with no further ado, let me introduce my colleague, Dr. Keith Flaherty from MGH and the ECOG Akron to move forward into the scientific underpinnings of the MATCH trial.
Thank you, Dr. Doroshow. Many people find ASCO to be breathtaking in terms of its scope and the clinical practice changing data. I find it voice-taking, so apologies for me for the croaking here on Monday morning. So I'd like to thank Dr. Doroshow and other NCI colleagues for their partnership with ECOG Akron, who I represent today and have been a career-long investigator within that group. I'm going to take just a few moments to set the stage for the scientific premise of NCI-MATCH. You've heard, I think, a fair amount even at this meeting, a really novel clinical science symposium organized yesterday to help try to address this issue of whether site of origin of a cancer still matters in the current era by which we consider cancers by their molecular or genetic definition. Let me start with just a few numbers to orient the discussion. 3 billion base pairs in the human genome, base pairs being those nucleotides, those individual molecular elements that are essentially the alphabet soup, if you will, organized into tens of thousands of genes and ultimately 20,000 genes that encode proteins, ultimately the doers within cells. As Dr. Doroshow and NCI colleagues had the vision to anticipate the potential benefit from decoding the cancer genome, it's that TCGA circle that he started with on the left-hand side of his slide that really began to give us a very complete catalog of all of the genes within the 20,000 that are common building blocks by which cancers assemble themselves. Based on present knowledge, about 300 or so genes within that 20,000 are the ones where one can find mutations frequently across cancers. But interestingly, there are about 200 different types of cancers. So 300 cancer genes, 200 different types of cancers. How do those sort, the cancer genes and the cancer types? Well, there are very rare instances of a cancer gene that sits within just one cancer type. You've heard of those examples. Chronic myelogenous leukemia is almost defined by a single genomic alteration. And the earliest targeted therapy success was using an inhibitor of the ABL kinase, the gene that is ultimately activated in chronic myelogenous leukemia, where that drug could be applied to patients essentially without any molecular testing, because you knew based on just the diagnosis that they had that genetic feature. Unfortunately, that is a very rare phenomenon. Actually, targeted therapy success number 2, gastrointestinal stromal tumor, has about 70% prevalence of the gene that is the target of the same drug, imatinib, that was the second breakthrough in the early 2000s. Already by the early 2000s, Dr. Huddis and colleagues were developing HER2-targeted therapy, but HER2 genetic alterations occur in only about 30% of breast cancers. And you've probably heard lots of deliberations about the relative inefficiency or maybe even impossibility of developing HER2-targeted therapy in breast cancer patients in an unselected way. So we face this realization that we need to hone down within cancer types or across cancer types at understanding the relevant genetic makeup or the building blocks, if you will. So when we start looking for those needles in haystacks, if you will, here within non-small cell lung cancer, you know these examples. When you look for EGFR mutations found in about 10, optimistically 15% of non-small cell lung cancer, and match those patients to erlotinib or gefitinib, you see reliable benefit from therapy, here measured by tumor regression on the so-called waterfall plot on the left-hand side of your figure. The empty rectangles being patients who don't have an EGFR mutation, the colored boxes being ones that do. This is a relatively older example, move into the 2010 to 2015 timeframe, and here you see an example with crizotinib being applied to non-small cell lung cancer patients who have the so-called ALK translocation. And this pattern of seeing reliable, not completely reliable, but fairly reliable early benefit from targeted therapy has been reproduced now multiple times. And we have perhaps now a dozen targeted therapy successes where we can claim successful matching, but generally within tumor type, as has been discussed. So my little corner of the cancer genome that I've had the greatest interest in is depicted here on this slide. BRAF mutations were discovered to occur in about 8% of all cancers, but distributed rather broadly across the cancer population. Melanoma, the tumor type that I treat, harbors these mutations about 50% of the time. Colorectal cancer about 8% of the time. Very common cancer though, so that's still a lot of BRAF mutant cancer cases. And there are a good many other cancers in which BRAF mutations have been discovered. But one of the moments that gave the field pause is depicted here. Here you see this exact same pattern that I've shown you in the EGFR and ALK instance, where a BRAF inhibitor applied to BRAF mutant melanoma patients produces this very same pattern of fairly reliable benefit at the population level just within melanoma. But swing over to colorectal cancer and look how unreliable BRAF inhibitors are. In fact, almost completely ineffective. Now, much of the motivation behind the MATCH trial is to look at both the left side and the right-hand side. Where can we find cancer genomes for which we have not only approved targeted therapies, but investigational targeted therapies as well? Deploy them across cancer, scoring successes when we reproduce this type of figure, and nominating those as opportunities to accelerate and pursue and ultimately approve those drugs for that patient population. But at the same time, where can we uncover the right-hand finding? This situation where the mutation is present, it's being targeted with the drug, but patients don't benefit. Can we understand more about what it is about context that matters? At this year's ASCO, just yesterday in fact, we've now seen the data that connects this clinical observation back to laboratory science, now back to patients, where combination therapy using BRAF inhibitor-based treatment with epidermal growth factor receptor antibody therapy is now able to impact these patients in a meaningful way and start to move them over to the types of outcomes that we're seeing with BRAF inhibitor monotherapy in the melanoma context. So we can learn about this issue of the comparative biology, if you will, of a drug being successful by itself in one context, unsuccessful in another, how to make it successful. This is ultimately the underpinning of the MATCH effort. The ERBB2 gene, again HER2, what I alluded to before, is still an example where we know about its benefits of targeting it in breast cancer. It's breast cancer where you most commonly find these alterations, either amplification or even more recently described mutations in ERBB2. But it's gastric cancer, bladder cancer, the next most common, and then cancers distributed out here on the right where you infrequently find them, but you still find them. We need to saturate all opportunity to apply therapies that we know can be beneficial against this target across the entire cancer population. And again, this is much of the motivator of the MATCH trial. The objective is defined here, to understand the relative efficacy of the same therapy applied to oncogene-defined subsets across the entire cancer population as defined by site of origin or tumor histology. And we propose to initiate a broad-based genomic pre-screening study. Dr. Conley will detail the operational characteristics of that in just a moment, to assign patients whose tumors harbor specific molecular abnormalities, assigning them to the relevant targeted treatments regardless of tumor histology type. This is the beginning, not the end, in terms of how it is that we think about applying these therapies, but it's ultimately through this approach that we're going to understand not only the relevance, but how to proceed and build ultimately rational combination therapies for this patient population. Thank you. Sorry, I'll just flash up as I leave here some of the design features. Dr. Conley is going to go into much more of the moving parts. We do need to test a large number of patients to find many of these genetic alterations. We're starting at a scale of several thousand that we're targeting, but this will be an evolving trial. We're quite sure as we go. We do need to biopsy tumors at the beginning of treatment. We've learned from many instances that if you rely on years old or even decades old archival surgical tumor specimens, those are not always representative of the tumor that you're in fact treating at the time of metastatic disease. You've already heard this is across the NCTN. We will look at tumor regression as the primary outcome measure. We are launching the trial with 10 arms, but we have a couple dozen more already moving towards launch, and this will be an evolving trial that adds new arms very much in a rolling fashion. We have representation and leadership from across the entire NCTN. We at ECOG-ACRIN have had the great privilege of playing a primary co-development role with the NCI, but as we move into the execution phase, we absolutely aim to work in complete harmony with the entire NCTN. It has taken an absolute village to build this trial. It has been hundreds of people who've supported the launch of the trial so far. Ultimately, it will take thousands of investigators even to execute this study. So this is a truly national effort. Very grateful for the NCI for promulgating it in the first place. And with that, Dr. Conley.
Thanks.
Okay, thank you, Keith. As you've heard, this is the largest effort of this kind ever initiated in the United States. We will have multiple treatments targeted toward molecular abnormalities, and we do expect the first patient probably to enter in July, and starting with 10 treatments, again ramping up to more than 20 within months. This will be conducted in peer-reviewed networks of high-quality clinical sites throughout the NCTN, academic as well as community practices, and patients who live in urban, suburban, and rural areas should be able to access this trial. It has been a highly coordinated effort. I know Keith went through that there are many people involved in the generation of this trial, but one of the key things we tried to do is go where the expertise is, get that expertise, and employ it in the MATCH trial. Okay, so, I'm going backwards. No wonder it looks familiar. Okay, here are the initial 10 arms that are going up, as you can see, crizotinib for ALK and ROS, those are both approved. Dabrafenib and trametinib are approved in melanoma for V600E and K BRAF mutations, but will be employed outside of melanoma in the trial. Trametinib for BRAF fusions, non-V600E mutations, non-V600K BRAF mutations. Afatinib for EGFR activating mutations and HER2 activating mutations. AZD9291 for T790M mutations and rare EGFR activating mutations. TDM1 for HER2 amplification. An investigational drug, VS6063 for NF2 loss, and sunitinib for c-KIT. And we will proceed with our molecularly defined groups to attain more treatments. I'll keep doing the other one, okay. So one of the important things that was mentioned is the diagnostics that will be employed in this trial. It's essential that the eligibility assays accurately identify patients with the appropriate molecular features in their tumor, and just as importantly, not to identify patients that they have a mutation if they don't. So we will start with a screening of 3,000 patients, kind of estimating that maybe a third of those will have mutations allowing treatment in MATCH. We do have in place a central biorepository and reference laboratory for the ECOG-ACRIN at the MD Anderson Cancer Center, and they will receive all of the samples to ensure the quality control. We have procedures in place for sample collection and shipping to make it very easy. We have a precise and reproducible next-generation sequencing assay and locked procedures to ensure reliability, and patients, NCI will fund the biopsies as well as the sequencing tests for this trial. Additionally, we have a CLIA lab network to perform these assays because our turnaround time is estimated to be 14 days or less. So we need to have a lab network in place to perform the study. The platform was chosen deliberately and with information to be the Ion Torrent Personal Genome Machine custom panel that looks at 143 genes with over 4,000 variants on those genes. It was developed at the NCI Frederick National Laboratory for Cancer Research by Mickey Williams and his team, who's in the audience. The labs that will be participating in running this is Massachusetts General Hospital, the Frederick Lab, the University of Texas MD Anderson Cancer Center, as well as Yale University. And just to wind up, the MATCH, of course, is part of the NCI Precision Medicine Clinical Trials effort, which also includes Alchemist, which is an adjuvant study, phase 3 randomized in non-squamous non-small cell lung cancer, which is already in progress. Lung MAP, which is a phase 2/3 with the potential for registration, randomized trial in second-line squamous lung cancer, which is in progress. MPACT, which is a little bit earlier in the evidence gathering for actionable mutations, a 700-patient pilot in refractory solid tumors, which is already in progress. And from the other end of the spectrum, the Exceptional Responders Initiative, where we obtain specimens from patients who did have exceptional responses from any systemic treatment, and that is also in progress. This is just a nice picture of the NCTN clinical sites demonstrating the reach of the NCI-supported clinical trials networks. And with that, I thank you for your attention.